Welcome to another episode of Science with Spice Between Experiments. I am your host, Dr. Danielle Spice, a PhD molecular biologist and biotech scientist. On Science with Spice Between Experiments, we talk to really cool scientists and ask them to break down their science in a way that is understandable to everyday people. I'm super excited about this episode because we got an opportunity to talk to one of the authors of a paper that is blowing up in the scientific community right now. We're going to talk to a PhD candidate in biochemistry from Stanford University named Carlo Armijo. And he is one of the authors on a new science publication talking about bacterial defense mechanisms against bacteriophages, which are viruses that specifically infect bacteria. And this new mechanism that was discovered that shows that a DNA strand can be made from a protein. We get into that. We get into this really cool process that he's working on right now in a different lab called UFMylation. And I'm really excited for us to get into it and for you guys to hear it. Hi, everyone. I am so excited to be able to sit down and talk with Carlo Armijo, who is a PhD candidate in biochemistry at Stanford University. And he's currently in the lab of Dr. Ron Capito. And he's studying, this is a bit of a mouthful, and we are going to get into it, called U-F-Mylation cycling and ribosome recycling at the endoplasmic reticulum. So why I've been so excited to introduce everybody to Carlo is he's actually a co-first author on a very recent science paper that is getting a ton of attention worldwide for work that he carried out in Alex Gayo's lab, also at Stanford. That in this paper, it's describing a bacterial immune response using a reverse transcriptase called DRT3. And let's just hang in there for a second because I'm going to give the science words for how this actually is described. Where in the science speak, it's a bacterial antiphage reverse transcriptase that uses two distinct reverse transcriptases and a non-coding RNA to synthesize alternating poly-GT8AC double-stranded DNA, which in non-science words, and hopefully, Carlo, you can tell me if this is totally wrong, but that's okay. This is how I interpreted this paper, is in these non-science words, it means that there's a protein that has the ability to make double-stranded DNA using some really cool mechanisms that don't follow how we typically think of DNA being made. And it does this as a bacteria trying to fight off an infection. Don't worry, everybody who's watching and listening, we are going to get into it, and we have an expert who's going to help us. But it's not all about the science. So before Stanford, Carlo actually spent 3 years at January AI developing microbiome-targeted nutraceuticals for metabolic health. And he's actually a named inventor on a patent, which is so cool. And he earned his BS in biochemistry from Santa Clara University. And outside of the lab, and I definitely have questions about this, Carlo, he is a professional pianist, a vocalist, an avid cook, and loves to spend time outdoors. So, Carlo, you are so popular right now, and I'm so stoked to talk to you. You're in the fourth year of your PhD. How is it going?
No, thank you for having me, Danielle. Yeah, appreciate this opportunity. No, it's PhD is definitely a journey, as you can attest to yourself. Just a lot of ups and downs of both science, like working and then not working, in addition to the stress of the science, and also dealing with different PIs and talking to different experts about how can you rethink about the projects that you're working on. I don't know. It's kind of like a nice, everyone calls it a pressure cooker, but I think as long as you sprinkle in it like de-stressors and really rely on friendships that you make, that's basically the only reason I'm alive. Kind of lucked out in both Alex's lab and now in Ron's. So, yeah.
So, have you lived in California your whole life? You were at Santa Clara before. Is California your home?
Yeah, no, I grew up in the Santa Cruz Mountains, as a little mountain kid, and pretty much, yeah, I went through high school and then through college, I stayed at home to save money. And it's been hard for me to leave. I mean, there's so many opportunities. California is amazing. You can go to the beach one day you go to Tahoe, ski the next, all in the same season, by the way. And then
Mhm.
like go to Napa for like some wine tasting. And then there's so many beautiful hikes. Like it's never ending. So every time I consider like, yeah, should I move to the East Coast for school? I think I've just been really happy here. So yeah.
I mean, I feel like California is this really kind of almost perfect storm of the stuff that you want to do. Like you're at Stanford, like there's so many amazing academic institutions as well as industry institutions in California, as well as all of the amazing stuff that you just described. I mean, I've only been surfing a handful of times, but it was very fun and I definitely love a hike.
No, I mean like the opportunity is pretty crazy here. And
Yeah.
like one day you could be talking to like a very like academic, like Nobel Prize winner, like Carolyn Bertozzi or something. And then the next you could be talking to like VP from Novartis or AbbVie or what have you. And it's really cool. It's a good like handshake. And I think the California vibe actually promotes like cooperativity and collaboration between not just industry and academia, but just in general science. But I love it here because no one ever like kind of turns you away when you have a
Mhm.
question. Yeah, everyone's super helpful.
So originally you were studying defense mechanisms in bacteria against viruses. And I really kind of want to unpack this for watchers and listeners because I would imagine if we're thinking about viruses, like we think about us getting the common cold or, even COVID more recently, are these viruses that are infecting bacteria the same as the viruses that we would get as humans or are they like totally different?
Yeah, I mean, so viruses in general kind of have like the main kind synonymous thing between the viruses that infect bacteria versus us is that they require a host to replicate. So basically turning either our human cells or bacterial cells into zombies. So that's where they're similar. Where they're a little different is that bacteria are a lot smaller than our own cells. Like if you imagine like scale, like a bacterial cell is either like a tenth of a human cell to a hundredth of a human cell.
Hmm.
Not only that, our cells are actually pretty fragile. They're just surrounded by a fatty membrane layer around it. And a lot of bacteria actually have two membranes. And within those membranes, they have like basically barriers, like walls of
Hmm.
sugars that keep it nice and protected. So the viruses that infect bacteria have evolved to really be able to puncture both of the membranes and release all their DNA inside. Compared to our viruses, those ones kind of float to our cells, and our cell actually absorbs it and takes it up. And that's how that works. But yeah, if anyone's interested in bacterial viruses, just look up bacteriophage, and you can kind of see
Mm.
it's a very interesting like shape. They look like robots. It's kind of like this like we call it the capsid, is like the shell where all the DNA and proteins are. And then it has like a basically like a siphon looking thing and like some legs. And what that does is the legs will sit on the bacteria and then it'll literally inject like a needle through their cell membranes and walls, and it'll kind of inject its DNA. It's
It's
kind of a neat
it's
thing.
it sounds like bacterial viruses are almost more violent than our viruses. They're like actually forcefully putting their genetic information into the cell versus for us, it's just like blub blub. Now we're infected with the virus.
100%. It is quite. You can actually look up videos online of and seeing how when the virus injects, like all of a sudden you'll notice the bacteria just explode. So not only
Oh
does it
boy.
inject, the bacteria just like yeah, it's kind
Oh
of cool,
no. Okay.
a little violent.
When we get infected with a virus, our immune system gets turned on. But when we're thinking about our own immune systems, we have our innate as well as adaptive immune system. And it's very, very complicated. Like the entire field of immunology is just thinking about the immune system. But if viruses also infect bacteria, then they must have their own immune processes. But I can't imagine they're the same as ours because we have multiple different cell types, but a bacteria is only a single cell. So how do they actually do that?
Yeah, that's a great question. Where we as like eukaryotes or mammalian cells kind of can rely on our whole body to develop a system-wide immune system. Like folks have heard of like antibodies and immune cells that come in, notice that a cell is infected and gets rid of it. Bacteria don't have this kind of level of communication or organization.
Hmm.
So they kind of have to internalize all of their adaptive as well as their innate immune systems. The other kind of factor that makes bacterial defense systems very unique is that the scale that both the viruses that infect them and the bacteria evolve is so much higher than in humans. Bacteria divide every 20 minutes compared to like a mammalian cell, like in our bodies. Sometimes like neurons never divide after, birth,
Yeah.
basically. Bacteria really had to develop to diversify their immune systems and evolve over time because it's an arms race between the viruses and the bacteria. Because if the viruses get too strong, there's no bacteria to eat. And so the
Hmm.
bacteria have to adapt. And it's, they call this the Red Queen hypothesis. But yeah, so typically how the immune systems work basically is like, so a virus is made of a protein and DNA. When it injects those things into the cell, the bacteria has to recognize that, hey, this is foreign to me. This DNA doesn't belong to me. And so many folks have heard of CRISPR, where specific sequences from viral DNA is what triggers the system in
Hmm.
bacteria. And what that does is it looks for viral DNA and chops it up the next time it sees it. The closest homologue to humans is like our antibody system. But for times where the virus kind of evades it, because the virus also adapts, it has kind of, in a way, simplified its genome or the proteins
Hmm.
that make up it, such that like most of the bacterial surveillance systems can't notice it. So bacteria just have to evolve new ways to notice that it's a virus and then decide, okay, is this something that I can kill the virus itself? I can chop up the viral DNA. If I can't, what it actually does is it actually commits suicide. So there's a point where the bacteria will decide, hey, I'm infected. I don't want my neighboring bacteria, my buddies, to die. So it's kind of called a programmed cell death. And it'll basically stop the virus from replicating inside the cell.
Yeah, so that's really interesting that I wouldn't have expected a bacteria that's a single celled organism to undergo programmed cell death to save its neighbors once it's been infected with a virus. I would think as a single celled organism, you would want to try to stay alive as much as you can, and you wouldn't necessarily care about the rest of the culture. Am I wrong in having assumed that? Like, it sounds like it.
No, it's kind of interesting. Kind of immune systems in bacteria, 90% of them plus go through this programmed cell death pathway, and you can kind of think about their neighbors less like, oh, they're individual people, because bacteria, for the most part, reproduce asexually, which literally means that one organism kind of splits off into two, and those are basically identical clones. So if you imagine you're cloning yourself in a room, and one of us gets sick, you're like, okay, well, there's enough of me around. If you could please like sequester yourself. I mean,
Uh-huh.
cell death, it'll prevent the rest of us from getting sick.
No, that makes perfect sense. So, you're, as I mentioned, one of the authors on this really fantastic paper. And I'm not going to lie, I was impressed with how much work you guys were able to put in a not very long manuscript. So, I definitely appreciate that. And like I talked about, it's published in Science, this internationally world renowned, respected publication, and it's getting a ton of attention from both the scientific community but also the non-scientific community. One of my personal videos on my Instagram actually blew up talking about this, and I'm not the only one who's had like tens of thousands if not hundreds of thousands of views talking about your work, which, I mean, great for me but great for you guys too. So why do you think everyone's so excited about this new research?
Well, yeah. So the lab can't take full credit probably for how it blew up. This was actually kind of the science editor's way to grab attention to this paper, by saying that we're breaking down the central dogma, right? Where everyone in biology class in high school knows that DNA codes for RNA, which codes for protein. And so at first glance, folks see like, oh, this is a protein that like skips, it doesn't even go through RNA to DNA, it just completely skips and like makes DNA, which is so backwards from what we learned. And so, I mean, to be like kind of grounded and humble and kind of like how we uh, discovered this protein, like it really isn't conferring genetic information from the protein to the DNA. So that's where I think a little bit of the science editors and the folks talking about this have kind of was like, oh, okay, this is kind of like some sneaky, interesting, like science stuff. That being said, it is the first instance where we do see a protein actually have specific DNA that it makes just from its own template. of its amino acid residues. There have already been, we call them DNA polymerases. Polymerase just is one word for when you make like a polymer of a certain molecule, and a DNA polymerase is a protein that synthesizes DNA. Typically, the proteins that say replicate in our own cells are all guided by our own genome. So it's DNA to DNA, but it requires that protein to read that template strand and write the new clone. In this case, though, why I think it's gotten so much attention is this protein doesn't have a template and it just creates DNA de novo. Not only that, that DNA has a specific pattern or sequence.
I think that is so cool. And I agree. And I'll call myself out on it. I could have maybe been a little bit more clear in my own video when I talked about this work, in that I think we were all just so excited as scientists to see this de novo from nothing, no template, being able to make DNA, that some of us, including myself, maybe lost some of the nuance to bring it back down to earth. So I'm really glad that you did that for us. But I think the real, like, so cool takeaway from my perspective is the idea that we're not just making random nucleotides. Because correct me if I'm wrong, my understanding of the field currently is that there are proteins that have the ability to string nucleotides together, but they're not specific. Is that true?
Yes, exactly. Alex and his previous colleagues, what they did is, so they looked for what are called defense islands in bacterial genomes. And what those are is basically the immune systems of cells. And those are horizontally gene transferred from one species of bacteria to another. So these are very highly evolving genes. And of that, there's a class of reverse transcriptases that is highly divergent. Um, I like 75% of all of the genomic diversity of reverse transcriptases is in this family. Um, and of that, there's a subclass that do actually make random DNA from no template. And this
Hmm.
kind of makes sense. They basically have the catalytic sites of a normal DNA polymerase, which means that it can grab nucleotides floating around in the cell and just kind of string them along. And so this has been observed in like 3 different proteins. And so this DRT3 system is the first case where the structure of the protein itself constrains the catalytic pocket. So now not every single nucleotide can fit in, only the specific A and C can fit into the DRT3B protein. And that's how it kind of has that pattern.
To me, that was the thing that was the such a cool breakthrough. Like you mentioned, that we're constraining the specific nucleotide, specific DNA sequence, even if it's not very fancy. It's a pretty basic repeating sequence, but it's using hydrogen bonding through the amino acid to call the correct nucleotide. And for watchers and listeners who don't know about hydrogen bonding, basically, usually when we're making a templated DNA strand, our polymerases don't necessarily know specifically if this is, an A base versus a T base versus a G base. It's all based off of chemistry and understanding what are the available bonds that can occur between these two different nucleotides that are coming in to do that pairing that we have with a DNA. So when we're thinking about this, D structure, this is the thing that's seemingly the most important for this. But as a biochemist, 3D structure feels just like important in general. So, how, in your experience, how do you even look at something that is that small? Like, it's not only microscopic, like down the microscope, but it's also like a single protein. Like, how do we resolve or break down what the 3D structure of a protein even looks like?
Yeah, no, that's a great question. So when proteins are so small that light basically can't resolve them. So that's why we can't look at proteins at a microscope. So we kind of rely on a technique called cryo-EM or electron microscopy. And the idea is so the cryo side is basically we have to freeze our protein sample down. And for that, you have to need a really pure sample of protein and lots of it. And basically you kind of put it on a grid, and it's the glass slide that you normally have in a microscope is actually replaced by water. Imagine just like a sheet of ice and all your proteins are all dotted on here. And these are like millions and millions of protein particles. And what the electron microscopy part does is that it shoots electrons through the ice. And if it hits your protein in just the right way, it'll actually bounce off of the electrons that make up your protein, and it kind of creates a shadow. So if you can imagine the sheet, if there's a protein here and you shoot an electron through it, it'll bounce off and you'll see a bunch of shadows pop up on the grid. And there's fancy technology that now even AI is improving also, that can take all those shadows and average millions of molecules together and kind of recreate a 3D structure from those images. It's really kind of groundbreaking technology, and I think it's basically better informing us how the individual molecules of life actually have an effect or cause disease, or we can discover so much about all these kind of unknown proteins this way.
So it sounds like the ability to have this single molecule resolution using cryo electron microscopy is super, super important for just us in general to understand function. And can you give any specific examples or just anything that gets you excited about how we can move any field forward, I suppose, or like medicine or whatever, when we have this such small resolution?
No, 100%. So I guess historically, there's kind of been a wave of scientists moving away from actual biochemistry and now focusing on genetic manipulations like CRISPR. And when that came out, folks, in order to understand a disease, is they would knock out a gene or change it somehow and basically see, oh, do the cells look healthy? Do they look sick? But now I think there's a new wave of actually going back to looking at the proteins that cause the disease rather
Mhm.
than just knocking out that whole protein itself and seeing what happens. So a good example of this is actually TR. It's a cystic fibrosis chloride channel. What is interesting, so this channel sits on your cell membrane and basically modulates the amount of chloride ions that go into and out of the cell. And if that's dysregulated, what happens is you get like really like thick mucus and bacterial growth in your lungs, and that's it's a really bad disease. But what scientists have done is they've solved the cryostructure of the disease version of this protein versus the healthy, and they found that it is like misfolded in the disease state.
Mm.
And they could only figure that out by looking at the actual structure. Not only that, is that when they added these new drugs, basically they resolved the structure when they added the drugs, and they showed that the whole molecule is refolded properly. Not only that, they saw where the drugs could bind in the protein. So there are certain diseases that you can't really understand unless you actually see the protein that causes that disease.
Wow, if anybody ever wanted to think about what they could possibly do to get us more forward in the field of medicine, actually understanding how like small molecule inhibitors or activators, how they like actually work, like the root cause of their actual function feels so important. I feel like watchers and listeners might be legitimately shocked by the number of drugs that we have that are fully approved, and everyone in medicine and science is like, well, it just does this. And everyone's like, but how does it do it? And they're like, I don't know.
Exactly. Yeah, they stop at the money making phase. The problem though is then the edge cases, right? Where it doesn't work. That's where really understanding the mechanisms can help us like not just save 80% of people, but now all
Hmm.
100%. So
And CFTR is a really great example of that because not everybody has the exact same mutation that is going to give them that exact same misfolding.
exactly. Yeah.
So I'm really interested as well by what you're kind of more recently working on. So what exactly is this UFMylation, and why is that important for ribosome recycling? So for anybody who's watching, listening, doesn't know what a ribosome is, your ribosome is what makes your proteins, and specifically at the endoplasmic reticulum. I feel like everybody can remember like way back in high school, elementary school, the rough endoplasmic reticulum. So it's rough because that's where ribosomes sit to help us make proteins that are supposed to be shuttled to different organelles or outside of the cell, if we're talking about a eukaryotic cell. So what is this new type of cycling? Tell us about it.
No, there's actually a joke in our lab that the hardest part about UFMylation is not the science, it's rather how you pronounce it. Because everyone says like UMFMylation, UFMylation, but it doesn't really matter how you
Oh my
say
God,
it,
no,
but
teach us, teach
UFMylation.
us. How do we say it properly?
The easiest way to say it is just UFMylation. So like UF and mylation. Yeah.
U-F-Mylation. I love that. If only I need to like go back and retell your bio because that's a way better way of saying it.
It's perfect. Yeah. So, like you were saying, the ribosomes kind of serve as the factory of all in the cell of all of the proteins that make us up. The ribosomes that go to rough ER, the rough endoplasmic reticulum, are synthesizing either proteins that will sit on the membrane of your cell. So, these are ones that are in charge of like communicating cell to cell, or secreted ones like hormones or different small protein signals that get sent throughout your whole body. Uffmylation helps those secreted proteins actually get properly translocated into the endoplasmic reticulum, which we can think of as like a delivery system.
Mhm.
You're almost like placing the factory right on the FedEx that's going to ship it out to the rest of the body. It's really been found that uffmylation is required in order for this process to happen efficiently and not have misfolded proteins. Because what happens when you have misfolded proteins is your cells can't really, what happens is the misfolded proteins actually can aggregate. It can cause issues. So some of us have heard about like tau, which is related to Alzheimer's disease. It's a protein that aggregates when it's misfolded and causes these neurodegenerative diseases. And tau has actually been really closely linked to the uffmylation cycle. When you shut off this cycle, tau aggregates like crazy, and we could basically get phenotypes that suggest Alzheimer's disease and different things like that. But what it is, it's actually, so UFM1 is this really itty bitty protein that compared to a giant ribosome gets plugged onto the ribosome after it's done we call it translocation. So the ribosome makes the protein in the factory and immediately like injects it into the endoplasmic reticulum through a portal that is called the translocon. Once that is done, the ribosome can't actually float away. It's actually stuck to the translocon. Biology in us has evolved to make sure that the ribosome doesn't float away until it's completely finished. And UFM1 is this tiny protein that comes and tells basically this uffmylation system that, hey, this ribosome is done translating. Now it can pop off and float away. And it's really important because we also think that there are some quality control features that once the ribosome is done translating its protein, and then while it's stuck, there's some features that come in and check the ribosome, make sure it's still a good ribosome and can be recycled to become a new factory for other proteins. Yeah, that's essentially why uffmylation is important. But it's what I'm studying is kind of the one of the main stages of that cycle, basically the final one that helps the ribosome actually come off of the uffmylation trap.
So, for anybody who is not hardcore into molecular biology and biochemistry, I just want to congratulate Carlo because describing ribosome recycling at the endoplasmic reticulum and how we actually get proteins into the endoplasmic reticulum is not easy. In a way that is not just like word vomit science words that are ginormous that nobody understands. So thank you very much. That was amazing. So I'm so intrigued by this and I'm flabbergasted that I didn't already know about it. Is this like a super well known part of translocating proteins into the ribosome? Or can you tell us, like, was it discovered a little bit more recently? Is that why you're so excited to work on it?
So it's been known that there has to have been some factor that helps these
Hmm.
ribosomes come off of the ER and recycle. And so Gunther Blobel is kind of the founder of the field of ribosomes at the ER. And for the longest time, he postulated that there has to be something because if you don't have like a wedge or something to drive between the ribosome and the translocon, it doesn't come off. So it was actually recently, and thanks to another cryo-EM solution by our lab in 2022, so this is pretty recent, we saw that the machinery that checks that the ribosome is empty and is also the same machinery that plops UFM1 on. Not only that, is that whole structure moonlights as the actual wedge. that puts itself between the translocon. So it's both a writer of UFM1, a reader of UFM1, and my project is now suggesting that a piece of this is also the eraser. So it takes UFM1 off and then the ribosome's free to go. The problem with the field right now is I think because it's hot, everyone's trying to find it correlating to different diseases.
Hmm.
Know that it's definitely related to tauopathies, but people are tying it to autophagy and a bunch of science jargon. But the problem there is that it's not really, we don't understand the mechanisms for how all of those diseases happen. And because of that, I think people are jumping to a lot of conclusions that UFMylation is happening to other proteins when really its sole job is to recycle ribosomes. And when you don't recycle ribosomes properly, the whole cell, like so many pathways start going awry and the cell can't handle it.
So, just because I'm so incredibly curious, are there any in the process experiments that you're able to share with us that you're excited about what the results might give? Nothing is published yet. And if you can't say specifics, that is totally understandable. Because, like you said, this is a hot field. So, you don't want to have anybody scoop you.
Yeah, no, unfortunately, yeah, this what I'm working on, I know is getting worked on in a couple other labs as well. Um, I have some right now. What I'm working with is a lot of cell-based assays. And so, like I mentioned before, with like the CRISPR knockout kind of experiments, that's what I'm really doing. Is I'm making a bunch of mutants of the components of the UFMylation cycle and seeing how these mutants actually affect how efficient the ribosome can get recycled or not. And now I'm actually moving towards going back to my biochemistry roots and actually purifying these proteins and seeing if I can manipulate them in a way that I can understand how quick each of them work. So we call this kinetics, since if basically each piece doesn't move as a well-oiled machine, the UFMylation cycle is kind of dead and your ribosomes accumulate, you get aggregated protein, and then your cell can't handle itself. Yeah, I think it's a huge open field, and there's kind of like some basic science questions that I'm able to do. And yeah, using a lot of AlphaFold to kind of help me design a lot of these mutants.
That's awesome. I feel like as someone who is like, a biotech scientist, maybe a little bit more translational, I can't wait until you get all these experiments done, publish it, so then people can then start working on like preclinical mouse models to see if there's any like drugs that could kind of help with the whole accumulation cycle. That sounds awesome. I'd
Uh,
be I'd be curious to know whether or not you could even do like a full knockout mouse of one of the components. It
well,
feels like it's too important. I think a mouse would probably not survive.
you're 100% correct. That was kind of the 15-year ago like goal of the lab, is to just kind of knock out these things. And in all cases, the mouse ended up not making it past a certain stage. There are a few like rare disease variants in humans, and a lot of those folks have a lot of like muscle or bone like development issues, and they can't survive till they're older. So
Hmm.
it's a super super important system, and it has to be like tightly regulated. And so we're figuring out ways that we can kind of know how it's regulated. And then potentially, actually, there are, at least I'm thinking about some translational aspects with some of these mutants could actually potentially be targets for industry, but we'll see if that's
That's
the route.
good though. I was gonna say, I feel like my next question was gonna be like, where do we see ourselves? I feel like it's a bit rude to ask like when you're gonna be done. No one should ever ask a PhD student when they're gonna be done. I feel like people ask me that for years, and I was like, I have no idea. I'm done when I'm done. But when you're looking at your future, I mean, you've already spent time with like an AI company, and now you went back to school to get this PhD. You've done a ton, and you've been so successful so far. Do you see yourself doing a postdoc next, maybe, or going into industry, or just kind of keeping our doors open?
Yeah, that's a it's a great question that it changes pretty much every day. But I think the more recent development is that if some of this research pans out, I think it would be nice to think about a startup and develop these in a more translational way. My schooling is all pretty much basic science, both the DRT system and this alpha-mylation cycle. But The UFMylation cycle actually has a lot of levers that I think could be usable. And so kind of chatting with a couple of my friends, my Stanford startup friends, we're kind of thinking about that. But yeah, for a while, like I was really thinking about postdoc and going the academic route, but I think this part is a little exciting to me. So we'll see
I
how it
mean,
goes.
having come from startup life, that's like my only industry experience is startups. So I can only recommend it. They're really fun. So I think you have a ton of potential opportunities in front of you, which is so exciting. I mean, I love just like living vicariously through, grad students these days now that I'm an old hack. I'm just like, oh my gosh, look at all these doors in front of you. I love it.
Well, that'll be me in hopefully a year and a half if everything works out.
Nice. Yeah, it's always the if, right? So I
Yeah.
find it's also like super important, for everyone who's watching and listening to just really show that us scientists, we're just regular folk. We're just like the rest of you. So outside of the lab, though, like when you take that lab coat off, what gets you going outside of the lab? What gets you excited? What makes you you that you're not a scientist?
Yeah, no. So the biggest other half of my life, I would say, is music. I've been kind of a professional musician or teacher. I used to teach marching band since college, basically. So it's been 10, plus years of that already. And now that's kind of my second gig, both to like help me with the grad school stipend, which isn't necessarily the best, but also like to give me a good outlet to just have a change of pace and like I really appreciate getting to kind of work with other musicians or vocalists that are so talented and we can kind of create like a beautiful sound, and it really takes me outside of all the stresses from the lab. And I think why I'm probably even leaning towards industry for science, because like collaboration and working with others definitely like really hones like an idea or a project, and that's what I really love. And then other than that, like people say I'm crazy for loving to cook, because it's basically like what I do in the lab too. I'm always pipetting, measuring, and everything. The trick that I say is that I'm a terrible baker because that's what I do in the lab. But then I just love having the freedom of like coming up with a new recipe.
Funnily enough, I feel like I'm the opposite. I'm an excellent baker. I am a terrible cook, but maybe that's just because I have one set of skills, they're lab skills, and they translate into the kitchen with baking. So funny. Oh my goodness. So I feel like you've had like all of these different like lives. Like that's so amazing. Like how multifaceted you are, Carlo. I'm so impressed by you. So I just wanted to thank you so much for coming on Science with Spice to, talk to us between your experiments and let us know what you're doing. That's so cool.
Oh, absolutely. Thank you so much for having me. Really appreciate the opportunity, and it's so nice to talk to you. Like it's good to know that there are scientists that care about communicating and like are super friendly. So I appreciate that.
It was so great to talk to Carlo about these new and exciting developments in biochemistry. UFMylation was so cool. This DRT3 reverse transcriptase is literally taking the world by storm. And I loved being able to sit down and chat with him. And hopefully now you all have a better appreciation for biochemistry and how amazing and cool it is. Don't forget that if you want to listen to more really exciting interviews with scientists breaking down their science, follow Science with Spice Between Experiments anywhere you get your podcasts or watch our conversations on YouTube. If you also want to follow me and get biotech updates and science concept mini lessons, make sure to follow me on Instagram and TikTok at Science with Spice. See you next time.